Next, we move on to Cakan. He is PhD researcher at the lab of Klaus Obermeier in Technical University Berlin. Over to you, Chala, please.
Sorry, I was muted. So thanks for everyone joining. I hope you can hear me. And so my talk will be kind of like an extension of what you've seen before. And let me just start with it. So here's a short outline. First, I'll talk about the interaction between neurons and electrical fields, how they interact in the first place. Then we'll present shortly a Meanfield neural mass model that can capture effects that we're interested in. And at the end of the day, I will show you how electrical stimulation affects a population model that I've just shown you. So let's go for first a little bit of motivation. Why are we interested in electrical stimulation? Probably all of us know that there are multiple stimulation techniques popping up in the last couple of years that are being very successful in treating diseases, DBS, TMS, and TES, for example. And most prominently, TES is a technique that is very easily applicable by placing electrodes onto the scalp. And these also have been verified through cadaver studies, for example, where people put the electrode out to the scalp and measure spikes inside. And you can see what you also might expect is that external fields, that's from the TES stimulation, can entrain spiking of the neurons inside of a brain, right? So in my mind, if you would want to model something like this, you would have to probably simulate a whole brain for that in order to know how an electrical field affects the brain dynamics. Another way to research electrical fields is to look at what happens in a slice preparation. So local populations of neurons usually put the slice between two electrode plates. And then here you see one example where the population is spiking at 25 Hertz, and then an input is turned on and off again. So you can turn on and off an oscillation. And if the input is oscillating, you can even have new oscillation frequencies emerging. And this you might simulate using a model of cortical patch, let's say excitatory and inhibitory neurons, and this is what I will talk about today. But let's go one step back first and see how do electrical fields interact with a single neuron. And for a pyramidal neuron, it's pretty clear that when you have an electrical field outside and you have ions flowing inside of this neuron, that the electrical field will affect this. So one way of modeling this would be a morphological model where you really place these tubes and cables and currents that interact with the electrical field. And the way this works is basically very similar to how an antenna works that we know and use every day. However, we like to use point neuron models, for example, AdEx neurons, and AdEx neurons don't have an extension, so you cannot couple an electrical field to an AdEx neuron because it doesn't have that spatial extension, so that won't work. And one way of solving this is from a colleague of mine was to fit a ball and stick neuron to the AdEx neuron, so a bit intricate. However, he calculated how the electrical field, Florian, affects the spiking of a ball and stick neuron and translated that into an equivalent input current to AdEx neurons. So it's kind of like getting an electrical field effect to point neurons, very shortly said. And so this is what I'll show you in the next couple of slides. And I've decided to not put any equations to how this mean field approach works, but just a image will suffice, I guess. So we have an AdEx network, delay coupled between E and I populations receiving inputs. And we do this mean field reduction that you can see in our paper, how it works. But at the end of the day, we get we do these typical mean field assumptions, and we get a population firing rate model that gives us the firing rate of the population depending on the mean inputs to both populations. So it's a linear, nonlinear cascade model that is extremely efficient, originally derived from a Fokker-Planck equation of these AdEx neurons. So we end up with a AdEx mean field model of AdEx neurons. And now I want to draw bifurcation diagrams of this model to see what it can do, right? So similarly to as the other talks that we've seen before. Now let's draw a bifurcation diagram in 2 dimensions. We have one input to the excitatory population and one input to the inhibitory population. These are the parameters that I'm changing, and you can see here the states that emerge. So in the inhibitory dominated regime, we have a down state where there's a low activity. In the excitatory dominated regime, there's an up state with high activity. These are fixed points. There is a bistable region where upstate and downstate can coexist, so you can jump between the two. And in the middle, here's a limit cycle in white contour where the activity oscillates between excitatory and inhibitory population. So this is the bifurcation diagram of the Meanfield model. And to verify this, we did also simulations, huge simulations with large networks of neurons to see if the bifurcation diagram is actually true. And it turned out to be quite true. So here you can see the analog bifurcation diagram of the AdEx network, now simulated with 50,000 neurons per point. So this is a very huge simulation that is going on. So just to, I mean, this guy takes about, let's say, 15 to 20 minutes to compute. This one probably a week or so on a live server. So we have adaptive neurons. This was without adaptation. Now adaptation, a single neuron for AdEx does usually this. So you have adaptation that makes the spiking slower as the input comes. So this is what it does for a single neuron. And we were wondering, what does it do for a population of neurons? So we turned on adaptation, and what happens was this. So the bifurcation diagram is transformed from the upper one to the lower one if you turn on adaptation. And most strikingly, what you see is that the bistable region now transforms into a new limit cycle. So you have now 2 limit cycles, one the fast one between E and I, and another one that is due to adaptation. So excitation and adaptation, basically. And let's look into these states. So basically, if you sit in one of these points, for example, you see a fast oscillation between E and I. And if you sit inside the adaptation limit cycle, you see slow oscillation that is due to adaptation, a lot slower than the EI oscillation. Okay, so this talk is about electrical stimulation. Let's talk about electrical stimulation. And one way, the reason why I also showed you these bifurcation diagrams is now we can try to understand what stimulation would do to these neurons, to this population. So imagine you're sitting somewhere in this bifurcation diagram, A1, for example, and you turn on electrical stimulation. It is equivalent to moving on the x-axis of this bifurcation diagram because we're changing the input to the excitatory population through stimulation. And why only excitatory population? Because, as I said before, you need extension for interaction with electrical field, and excitatory neurons have this antenna-like structure, pyramidal neurons, for example, but basket cells, for example, are much smaller and more symmetric, so you don't have much interaction with the electrical field. So this makes the electrical field input basically move on the x-axis here in these diagrams. So let's do now this. We put the bifurcation diagram to the side and then sit on this point A1 and just repeat what other people have done with the experiment. So for example, we start in the down state. Now we turn on the stimulus and we can see how we move from the down state into the limit cycle by a constant stimulus, right? So this turns on an oscillation through a direct current. The same is also true for the AdEx network. Of course, if you sit in an equivalent point in the bifurcation diagram. We can also turn off oscillations if you start in a limit cycle and then push it into the upstate. So you see the upstate here, for example. Same also for the AdEx network with a longer transient. We can also, this is how we detect the bistability in the AdEx network, for example, is by pushing it up, then you relax the stimulus. So you're in the upstate, and then you push it down and you relax the stimulus again. So the input here and here is the same, but the states are different. You see, here's an active state, here's an inactive state. This is how you can detect bistability, for example, in a spiking network. And you see the same also in the spiking neuron model. You can have phase locking. So if there's an oscillation going on and you input an oscillation, then you will have interaction of these oscillations. And you can also drive oscillations with an oscillatory stimulus. So I'll quickly talk a bit more about the oscillatory stimuli, what they can do. And now in this example, so the population is oscillating at 25 Hertz. Here you can see the frequency spectrum of the oscillation, and this is in the EI limit cycle. And now I'm turning on a stimulus with a specific frequency, and the stimulus frequency will change. This is a movie now. And here the frequency spectrum of this thing, you can see here is the peak of the oscillation, and in red you see the stimulus frequency. And now I will change the stimulus frequency. I want you to pay attention to what happens if the stimulus frequency approaches this peak. So something will happen and you can see what. So I'm changing the stimulus frequency now. There's lots of frequency going on, and now something happened and it dropped back to the original shape. I hope you catch what it was. So I'll just replay again, and you can see that when the stimulus comes close to this peak, it picks up the peak and moves along with the peak. Yeah. So this is called frequency entrainment, and we can measure this. So this is now the same diagram, but looked at once. Basically, we're changing the stimulus frequency on x-axis, and we're drawing the spectrum on the y-axis, and the color is the height of the spectrum. And you can see here the frequency is going on of the oscillation until it reaches this oscillation frequency, the resonant frequency, and then entrainment happens. So it takes up the peak and moves it along until it's too high, and it drops back into its original frequency. Now, since we have a biophysical model, we actually can measure what the electrical field strength of this will be, and this is around 1.5 volts per meter. We also did the same for the AdEx network, which is a slightly higher frequency as well. So I'm a little bit out of time. We also did the same with higher frequencies, and you can see all sorts of interactions of frequencies going on here. And so we measured frequency entrainment and measured also the field strengths. We also measured phase locking, because this is important for stimulation experiments. And what I can tell you is that phase locking also works in this population model with much lower electrical amplitudes. So my conclusion very quick is mean field approximations work very well if you want to speed up your simulation time. Also, non-stationary interactions are well captured. DC input can, for example, cause attractor switching. AC can frequency and phase entrain. And most importantly, the field strengths that people use in experiments really seem to have an effect on the population activity. So this was my talk. You can simulate everything also using our framework here, NeuroLib, and thank you for the attention.
Thank you so much, Jalar. It was really a very fascinating talk. I think we have a question from Matteo. Please go ahead.
Okay, yes. So thank you. It was a very nice talk. So apart from, it seems that the Meanfield model works very well. I was wondering, of course, in different states of the brain, we have neuromodulation that changes. So for example, the strength of adaptation can change depending on the state. So my question is, how does the resonance frequency, or for example, the amplitude of stimulation that you need to change oscillation depend on the level of adaptation that can be its strength or the time scale, which can change, for example, in different states, as if your tool to switch could be state dependent in a certain sense.
Yes, so thanks for the question. This is indeed true. We also did lots of exploration on what changes in the bifurcation diagrams when you change certain parameters, for example, adaptation. So this was one slide that I didn't include, but you can see maybe, oops, I cannot show you the slides anymore, but I can tell you that changing adaptation also changes the bifurcation diagram. And the same is also captured in a mean field model. So that still works for changing parameters. And it is completely true that the state, it really depends on the state in which you are, how the reaction to the stimulus will be. I would even say it's even more important than the stimulus parameters in a sense. So the stimulus parameters might be 10 Hertz or 15 Hertz, but it really has an effect whether you're in a fixed point, for example, or in an oscillation already. So I can only tell you that there's a lot of dependency on the state, and it really depends on where you are in the state space.
It's very interesting. Thanks.
Thank you very much. I guess we are running short of time because the next session is